It's Wednesday, the 13th of October, 2021. But in Lake Tahoe, Nevada, it's still the 12th of October, and I'm talking with the legendary Bob Proulx, who's the CEO of Imagion Biosystems, ASX IBX. Bob, good afternoon.
Good afternoon, Stuart. Nice to see you again.
When you told me that you were living now in Lake Tahoe, I have to confess I broke the 10th commandment there briefly. But it's testament to the fact that the life sciences are truly global. Here's Imagion Biosystems, publicly traded on ASX, CEOs in Lake Tahoe. Your CFO is Jeff Hollis, is in Melbourne. You've got a laboratory in San Diego. Where else in the far-flung Imagion empire are we located?
That's pretty much it. Although we do now, having made some progress with our work, we have a few more people in Australia. So we've got a couple of business development people, one in Melbourne and one in Sydney. So we're expanding our capabilities in Australia while we still develop the product here in San Diego. And I'm currently taking advantage of the lovely coming winter here in the Lake Tahoe region.
Right. Now, many investors may have seen the article we wrote a couple of months ago on Imagion Biosystems, but Bob, refresh our memory. What Imagion Biosystems has one of the more powerful cancer diagnostics that's still coming. Describe to us the technology that you're building.
Yeah, thanks. So we're a medical device company developing what we consider to be sort of the next generation of non-invasive molecular imaging. So, you might remember that conventional imaging methods like ultrasound, CT, MRI, they're very good at visualizing tissue and identifying areas of suspicious lesions, but they're not very good, for example, at being able to differentiate benign from a malignant tumor. And so, even when they are able to identify a lesion, they're kind of limited in the size of the tumor that they can identify. They need about a 1 centimeter spot to be able to identify it. And typically in the growth phase of cancer, that starts to get late stage. By the time the tumor is about 1 centimeter, you've progressed pretty significantly. So our MagSense technology is using biosafe magnetic particles to tag the cancer. These particles have the property that the magnetic property of the particle allows them to act like a magnetic beacon when they become attached to the tumor. And so we are offering a very sensitive, specific way to non-invasively detect tumors without using radiation like what you'd use in a PET tracer.
Right. And arguably at low cost. I mean, we're all used to the expense items in the health budgets for MRIs. You're about to blast a hole right through that budget, ultimately.
Yeah, we're hoping to be able to do that. I mean, the equipment that is used for MagSense technology is less expensive than an MRI, doesn't require a shielded environment. Additionally, the injectable imaging agent is going to be able to be made at an affordable price. It has a long shelf life, unlike the use of a PET tracer that needs to be made typically in a local cyclotron and has a relatively short half-life. Our technology lends itself to being able to be something that actually can be packaged in a way to be more cost-effective when administered to the patient.
Right. Now, when I'm describing Imagion, the shorthand I use is 1,000 times more sensitive than MRI. Now, that's not an exaggeration, is it?
Well, we hope not, right? I mean, that's our aim here, right? So if you think about the ability for MRI to visualize a 1 centimeter tumor, we're looking to try to get that down to about a 1 millimeter tumor, similar to the sensitivity of a PET tracer, but without having to use a radioactive isotope to detect that. So yes, we think that we're a good order of magnitude or more sensitive than MRI would be, for example.
Right. Now, you and I first met about 2019. You've made a lot of progress since then. MagSense is now in the clinic. Admittedly not recruiting as quickly as you'd like, but hey, look, that's the times we live in.
Yeah, fair enough. Yes, we're very proud to have made it into the clinic. I mean, this is a major milestone for almost any biotech company, right? To get their product into the clinic, into first-in-human testing. So yes, earlier this year, we got into the clinic in Australia with our first clinical site. We enrolled our first patient in May. And as you well said, yes, it's been a little bit slower than we anticipated in terms of recruitment. But we've added a number of sites. So I'm very happy to be able to tell you today that our fourth site is now up and running. We've added the Lake Macquarie Private Hospital in New South Wales, not very far from where you are. Yep. This is part of the Ramsay Healthcare Group, one of Australia's largest private healthcare providers. And so now this gives us sites in the state of Victoria, in the state of New South Wales, and in Queensland. So we're finally in a position now where we think we can begin to expand that enrollment into our study. We were very limited at the beginning in only Victoria. And as you know, there's been significant lockdowns in Victoria, but we're anticipating to see that start to shift. New South Wales is coming out of lockdown and, for example, are reinstating breast screening services. So we think that we might start to see a change in our recruitment capabilities just now that Australia is finally getting a little bit of a handle on this. You may have seen, Stuart, there's been plenty of reports of reduced breast cancers, or not only breast cancer, but cancer screening in general. There's been a lot of worry about people going undiagnosed and the impact this might have. And so we're hopeful that with things hoping to return to some more measure of normality in Australia, that and now with 4 sites being able to start to recruit, we'll see an uptick in our ability to start to populate this study.
Right. So we're evaluating the technology to detect breast cancer. What's the exclusion criteria in this study, or inclusion and exclusion criteria?
Yeah, so that's one of the interesting things about this is that our first product is for a very particular form of breast cancer, HER2 expressing breast cancer. So that's a subset of all breast cancer patients, which also is part of the limitation in the number of patients that we can identify, right? We're only going after a particular form. And there's a reason for that, and that is because we knew that we could identify a very specific targeting ligand for our particles that was highly specific for HER2 breast cancer, right? And for our first product, we really wanted to make sure that we were going to achieve that level of specificity and sensitivity and not necessarily go for the home run, as we would say, or you would say, go for a 6, I guess, right? But we really wanted to get that base hit, demonstrate that the targeted nanoparticles could work. So, so our first study now is looking at metastatic breast cancer. Breast cancer patients, the first thing they want to know when they've been identified as having a tumor, they want to know, is it spread to the lymph nodes? And the way we do that today is by surgically removing the sentinel lymph node or by doing biopsies on those lymph nodes. That's great. That works really well, except for the fact that more than half of patients are node negative. So the doctor is going back to the patient and saying, I got good news, no metastatic spread. Bad news is I just cut your lymph nodes out to try to get that answer. So we saw that as being a real unmet medical need, that if we could non-invasively differentiate node negative patients from node positive patients, we have a way to improve healthcare for 50% of the patients. And in fact, the FDA in 2019 looked at our application for this HER2 breast cancer and said, if you can do that, this would be a breakthrough, because today we're having to do biopsies on every single patient. And so they designated.
When we wrote about that, we flagged the potential for breakthrough device designation down the track, right?
That's right. And so we got that. And so ultimately, when we do our follow-on studies here in the United States, we'll be doing that as a breakthrough device. But it's one of those things where limiting our focus on HER2 breast cancer isn't a bad thing from the perspective that we know we can look at a very specific outcome here. We know we're still addressing an unmet medical need. It reduces the target population for us to begin with, but it helps us, I think, from the clinical study perspective, because we'll be able to get very straightforward answers against the current standard of care of pathology.
Right. Now, for women who are watching this interview, presumably the study's up on clinicaltrials.gov and whatever the comparable websites are down here, right?
That's right. It's listed on the ANZ CTR, Australia, New Zealand. Yeah. So it's listed there. We've been doing a lot of outreach through social media, Facebook, LinkedIn, and other places, trying to get the word out that this study exists. And so, again, it's a somewhat limited patient population. It requires a newly diagnosed HER2 patient. And so, because we don't want to look at patients who are already undergoing treatment, because we don't want to interfere with the treatment, for example. But yes, there's an increasing visibility through our sites now and through some of the social media work that we've been doing to try to get the word out that this study exists in Australia. I mean, this is a first of its kind study. To our knowledge, no other manufacturer has ever brought a targeted magnetic nanoparticle into the clinic for the detection of cancer. And so this is big news, as far as I'm concerned, for Australia in general, and for us being a medical device, an ASX listed medical device company. We're doing something that's not ever been done before.
And what's more, admittedly, it's a little bit difficult to find women at that point, just when they've been diagnosed, but before the potential lymphodectomy. But let's face it, it's 1/5 of all breast cancer, roughly. Right. So HER2 is a not insignificant population to be testing in, right?
Well, not only is it not insignificant, but it's also one of the more aggressive forms of cancer.
Right.
So if you're worried about metastatic spread, whether it's gone to the lymph nodes, unfortunately, HER2 is one of the ones that will tend to be metastatic compared to some of the others. So it is really important that we're able to differentiate that. And look, even with pathology, there is known to be a false negative outcome, right? Sometimes in pathology, they miss it. So a patient is diagnosed as being node negative. They undergo surgical treatment of the primary tumor, but unbeknownst to the physician and the poor patient, you've got latent disease sitting in the lymph nodes that was never addressed. We're hoping to fix that. We're hoping to have a very highly sensitive test that can really differentiate node negative from node positive patients and say, those patients are truly negative, leave them alone, go ahead and treat the primary tumor, but leave the lymph nodes alone so that you don't end up having the side effects associated with lymphedema, et cetera, that you just described.
Bob, it's not just HER2 positive breast cancer. There's a whole pipeline of MagSense opportunities building out, and you've got collaborations with people like the CSIRO and ASX listed Patras PAB. Yeah. Talk to us about some of those initiatives you've kicked off.
Yeah, so as we got closer and closer of moving the HER2 product into the clinic, I wanted to be sure that we had a pipeline of products, right? We didn't want to be a so-called one-trick pony of just HER2 breast cancer. So we began working in earnest on our pipeline probably about a year and a half ago. But this past year, we're very fortunate. We got a small CSIRO grant to focus on prostate cancer, which we're now doing with folks at Monash University to help us with animal studies associated with that. We've been talking to the folks at Patras about their deoxymab product as a possible way to target brain cancer. And so this past year, we started to do that preclinical work. All the work that we had done with HER2 in the preclinic of doing in vitro and animal models, et cetera, we've now started to build that pipeline because I think we said to you as you covered us originally, our view has always been that the right way to commercialize the MagSense technology is for us to partner with someone. And whether that's a GE or a Siemens or a Toshiba or somebody, we think that they're already players in that medical imaging space. We'd like to partner with them. Well, we know that in order for us to be an attractive partner, we got to have a pipeline of products. They don't want to bring just one test to market. They're going to want to know that there's more behind that. So this past year has been all about getting the HER2 product into the clinic and establishing the fact that we've got more than one kind of imaging agent and that we can tackle multiple types of cancer.
So finally, Bob, for investors who don't know you, tell us a bit about your background. I like to joke that you get around the streets of Lake Tahoe and people say, are you the brother of the lady who wrote the shipping news, which I think we established that Annie Proulx is not your sister. Tell us about Bob Proulx, the biotech entrepreneur.
There you go. Thanks. So look, I've spent 30 plus years of my career working in the life science, instrumentation, medical device, diagnostic space. So almost all of it has been in trying to identify new and interesting technologies that could either improve laboratory medicine, laboratory research, et cetera. So I've always had sort of a very technology focus to my career, but mostly from the commercial side. So how do you define the product, the market needs? How do you bring it into the marketplace? I've led global sales organizations. I've established subsidiary operations in different countries, for example, to commercialize product. And so really what I think I brought to the table in joining Imagion 6+ years ago now was a commercialized focus to the business, right? It's not only about the technology. It's about what can you do with the technology and do you understand the path of how to bring that into the marketplace. And so I think we've been pretty rigorous about having a roadmap of identifying, and pragmatic, I should say, about how do we approach product development to demonstrate that we've got sort of the minimally viable product. We have a known unmet medical need. Clearly, that has been vetted by the FDA with their breakthrough designation, and that you have a clear understanding of the steps that you need to go through. And I would say that I'm very fortunate that in addition to my 30+ years of experience, we got an extraordinarily experienced board of directors. We have people who were the head of the chief technology officer at GE Healthcare, on our board, right? Every ultrasound, CT, et cetera. I think maybe I can't remember if you met Mike, for example, or not, but Mike's on our board of directors. We've got people like David Ludvigsen, who are serial CEOs and entrepreneurs developing medical devices and diagnostic products. Diane Angus, who's an Australian serial CEO of biotech companies. She's been, developed therapeutics. So, we're very fortunate that we've got a highly competent board with knowledge about not only the development of technologies, but how do you commercialize them? How do you properly fund a company, et cetera? So I think we're in good shape today. As I said, we're extraordinarily proud to have made it into the clinic. We can see what's in front of us. We're trying to get through that clinic, and we started to build the pipeline of other products. So I'm pretty optimistic about our next phase of our business.
Well, Prue, well done on everything you've achieved to date. I'm particularly pleased about the breakthrough designation from FDA, but it's executed magnificently. And if the hospitals in Victoria had come to the ballpark, we'd be even further down the track. But you've achieved a heck of a lot, so well done.
Great, thanks very much. Nice to catch up with you again, Stuart. Take care.